Okay, guys, so now we are on the third lecture for the neuron lecture. At this point, you should have already seen the reflex arc, the resting membrane potential video, and the graded and action potential video. Make sure you've already seen those up until this point. And now what we're going to do is we're going to talk about once you create an action potential, how do you propagate or move that action potential along the length of the axon so that you can send information from one part of the body to another part of the body. Okay, so we'll talk about how you get it all the way along, and then we'll talk about how you synapse or connect to a second cell in order to create a response in the second cell. So let's talk about those action potentials. So remember that action potentials are all or nothing. And what that means is once you start an action potential, once you reach the threshold to create an action potential, it's going to have exactly the same amplitude and behave exactly the same way. So one action potential cannot change. So if you're talking about propagating or moving the action potential, what you're really doing is creating a series of separate action potentials that are right next to each other. So this is very similar to like the domino effect. You knock over one domino, that'll knock over another one, and then another one, and then another one. But if you step back from it, it kind of looks like a fluid wave, right? So here we have an action potential in one specific location that will create the stimulus that allows it to reach the threshold to create an action potential of the place right next to it. And then while this area here repolarizes, that one will depolarize, and then it just sort of moves along a series in the series. So what I really like to think of it as like doing the wave, right? So if you have an action potential, you have the depolarization phase, becoming more positive, followed by the repolarization phase. So here's one action potential. But if they're all in a series, right, if you have a bunch of people all in a line, and you have one person doing the action potential, and that acts as the stimulus for the person next to it doing the action potential, if you all do it at the same way, you get the wave, which looks like a fluid movement along the length of the action potential. Right, so it's not, so the action potential itself is not moving. What it is a series of separate, different action potentials all next to each other. Right, so here's one single action potential and another one and then another one. Okay, so they just propagate along that way. And the speed that the action potentials are moved along the axon depends on two factors. The first is the diameter, and diameter is the distance across a circle, so how thick it is. Ones that are thicker are going to go faster, and that just has to do with when you have a bigger space, you have reduced resistance. And this is the same thing that also is for, like, if you think about in physics, talking about the spread of charges along, for example, a wire, thicker wires. So thicker just makes it so that there's reduced resistance, and the reduced resistance results in a faster conduction. I'm not going to ask you any more about that piece. I just need you to know that a bigger diameter is faster. But for myelination, you will need to understand more specifically. So when you have more myelination, that is faster. So let's talk about what myelination means. A little bit of anatomy review here. So here is our neuron in purple right here. Here's the cell body. These are the dendrites. Here is the axon. For some axons, depending on the type of neuron, some of those axons will be wrapped around by cells. Okay, so either Schwann cells or oligodendrocyte cells, depending on if you're in a peripheral nerve or a central nervous system. And this sheath that covers the axon, that wraps around the outside of the axon, that creates the myelin sheath. So myelination just means that the axon is wrapped around with those other kind of supporting cells. Okay, so then it stretches out and it wraps it. I like to think of it this way. So if you think of the axon like a wire moving information along, okay, you have a long wire, which is the axon. I tend to think of this sort of like the plastic insulation that surrounds that axon. Okay, so that helps, it does help protect the axon, helps it keep it nice and straight and protected. the other thing that it does is it increases the impulse speed. And this has to do with, when you think about that myelin sheath, it's not continuous. So this is why it's not like the plastic sheet on a wire. Okay, so it's as if you took that plastic sheet on the wire, but you left spaces between the sheath. Okay, so the spaces between the myelin sheath, between the cells, those are called the nodes of Ranvier. Okay, so the nodes of Ranvier. Those are just the spaces between the cells that make up the myelin sheath. Okay, so with that in mind, how do you think myelination increases the speed of the conduction? Okay, so maybe a next prompting question is, do you think you can create an action potential when there's a cell sitting on top of the axon? No, you cannot. Okay, so here's your axon right there. Here is your Schwann cell or oligodendrocyte. Here's the cell sitting on top of that that creates the myelin sheath. In the regions that are covered by those cells, you cannot create an action potential in those spots because the sodium and the potassium can't get through, right? It's covered, it's blocked. So instead what happens though is because you've created an action potential at one node, what happens is it gets to jump over that spot and then create an action potential at the second node. So instead of creating action potentials at every single place along the entire axon, you get to create fewer action potentials because you get to jump over the cells. So you're jumping from node to node to node. And because you generate fewer action potentials, it takes less time to go the same amount of distance because it's faster to go to make fewer action potentials. Okay, so you have fewer action potentials, and that's what increases the speed of the conduction. Right, so if you're thinking about a simulation where every single person makes one action potential, if you take so if you take maybe 5 people, and have those 5 people do their little action potential, and you create it and you timed it. If you had all 5 people doing their little action potential, that's going to take a longer time than if you just did every other person, right? So if you did all the 5 people, that's going to take a longer time than if you had only every other person, so maybe 3 people doing their little action potential. It's going to go much faster because you're generating fewer of those action potentials, and that's what increases the speed. So if you're looking at it, which axon would have the fastest speed? So I'm going to stop talking, let you look at it. Okay, so the first question we need to ask ourselves is, if you're trying to go faster, do you want a small or a large diameter? You want a large diameter so we can get rid of A and we can get rid of C. Okay, so now our choices are between B and D. Do we want myelinated or unmyelinated? Myelinated. So the correct answer would be D. A D allows for the fastest conduction speed because it is bigger and it is myelinated. So you get to jump up between those nodes. Okay, so now we've taken that signal and we've taken the action potentials and propagated, moved them, conducted them along the axon. How do we connect or talk to the second cell? That's called the synapse. Okay, so the synapse is the connection with another type of cell. Usually these are other neurons, especially what's happening in the central nervous system, but they can also be other types of effector cells such as glands or muscles, right? And we've already seen this before when we talked about the muscle lecture. Here would be the axon terminal of a motor neuron. Here is a skeletal muscle cell. So this would be a synapse between a neuron and the skeletal muscles. They also have things like glands, but usually what you're talking about is the axon terminal of one neuron connecting to the dendrite of a second neuron. That other cell is often called the postsynaptic cell. Post meaning after the synapse cell. Okay, so Though here in the axon terminals, those axon terminals are going to release chemicals, and those chemicals are called neurotransmitters. And neurotransmitters are very similar to hormones in that they are both chemical messengers. They are chemicals released from cells that cause other cells to do something, but instead of being released from glands, they're released from neurons. Other than that, they're very similar. Right? So they're a chemical that causes another cell, some other different cell, to create a response. Now, at this point, we've already seen one neurotransmitter, and the neurotransmitter that's released from somatic motor neurons is acetylcholine, so make sure you're very familiar with that. We're also going to talk about norepinephrine once we get to different divisions of the nervous system, so eventually you'll need to know norepinephrine as well. But at this point, you should definitely know acetylcholine. Okay, so let's talk about how you release the neurotransmitter. Here we have the axon terminal. Okay, here's the axon terminal. There's the space, which would be the synaptic cleft, and here would be the postsynaptic cell. So the action potentials are going to be a series of action potentials will travel along the axon. Once they reach the axon terminal, there's going to be a change in voltage, which will open voltage-gated calcium channels. Where have we seen calcium already? Remember? Calcium acts as the signal for muscle contraction. So you'll notice that calcium is often used as a signaling molecule. So here we have calcium, voltage-gated calcium channels opening. That's going to increase the concentration of calcium inside of the cell. So influx means going into the axon terminal, into the cell. What that does is it triggers exocytosis using those vesicles. So remember, in exocytosis, you have a vesicle that contains the product. In this case, the product would be the neurotransmitter that causes them to move towards the plasma membrane. Then because this membrane here is the same composition as the plasma membrane, it can just fuse right in with it. So to just sort of become one with it. And when it fuses with it, that means that anything that was inside of it will be dumped out. So in this case, the neurotransmitter will be released into the synaptic cleft. Now, here's another question for you. So at this point, we've just dumped a bunch of acetylcholine onto this side of the synaptic cleft. How does the neurotransmitter travel across to reach the other side of the synaptic cleft? What process allows the movement of the acetylcholine? Has to do with concentration gradients. We have diffusion. So all it does is diffuses it. Because there's a higher concentration of acetylcholine on this side than there is on this side, it'll just diffuse outwards. Okay, so it diffuses it outwards from high concentration to low concentration. Once it reaches the postsynaptic cleft, it's going to bind to a receptor that is specific, so lock and key type shape model. Bind to the specific receptor, depending on the shape, and that's going to trigger a response in the second cell. Okay, so don't forget about that role of the calcium. Calcium is often used as a signaling molecule. Okay, I hope that was helpful. I will see you later.